Epigenetic inheritance literally means inheritance that is on top of the genes. And so what that's referring to is that it's a modification of a gene or chromosome, not a mutation. So that's the key. It's a modification, not a mutation. In other words, not a change in the nucleotides. And this usually occurs during egg formation or sperm formation or very early in the embryo. And these modifications are going to change the gene expression for a way that is for the individual's lifetime. So it has an effect on the phenotype of the individual, but it's not necessarily passed on to future generations. And again, there's not an actual change in the DNA sequence. We're going to look at two examples of epigenetic inheritance that will help you understand this better. The first is X inactivation, and then we'll also talk about genomic imprinting. So X inactivation, like the name suggests, surrounds the X chromosome. So remember that we only need to have one X chromosome to be humans, but women have two. And so in our cells, we will actually shut off one of those X chromosomes. So only one X chromosome is active inside of our cells. So what will happen is during the embryonic development, one of the extra X chromosomes will be turned into a bar body. So here's the regular X chromosome in yellow, and notice how it's loose and readable. The other X chromosome will become very condensed, very tight. And remember, tight DNA can't be used. So this is a change that's happening to the X chromosome. It's still there. The genes are still there, but I've made it super tight. So this is epigenetic. I'm not changing those nucleotide sequences, but on top of those genes, I'm making it super tight, and so it's not usable. Only this X chromosome is usable. This is part of a broader concept of dosage compensation. Again, we only need one dose of the X chromosome to be human. But we have the two X chromosomes, so we need to get rid of one of them to get to the correct one dose. So X inactivation is a dosage compensation. It gets rid of the extra X so that we only have one X chromosome that's being used. So if an aneuploidy person is female but has two bar bodies in her cells, what sex chromosomes do you think are present in this individual? Remember, a normal female would only have one bar body. So this female probably is XXX. She has three X's, so two of them have to become bar bodies. This also explains why sex chromosome abnormalities are much more tolerated. Remember back to when we were talking about aneuploidy. Most of the sex chromosome aneuploidies don't cause very severe symptoms, and that's because of X inactivation. We only need one X. If we have extra X's, we'll shut them off. X inactivation is random, and this is really fascinating. So we don't always turn off one X chromosome or the other X chromosome. Some of our cells will use one of the X's, and some of our cells will use the other X. Because the X you inherited from your mother and the X you inherited from your father are virtually identical, you usually can't tell. But in cats, orange and black coat color is on the X chromosome. So there's obviously more than orange and black coat colors in cats, but just the orange and the black are on the X chromosome. So if a female cat is heterozygous, she has an X carrying the orange allele and an X carrying the black allele. Some of her cells will make a bar body out of the black allele, but other of her cells will make a bar body off of the orange allele. So this cell can give rise to orange pigmented fur. This cell would give rise to black pigmented fur. So you see patches of black and orange because the different X chromosomes are randomly inactivated. This is called being a mosaic. Females are a mosaic for their different X chromosomes because they randomly shut off one or the other. There's actually a trait in humans, our sweat glands, is controlled on the X chromosome. And so the females who are heterozygous for not having sweat glands, this is a rare condition, it's not common, but if they're heterozygous, they'll have patches of skin that have sweat glands and patches that don't. Again, because we randomly turn off the X chromosomes. So you discover a calico male cat. What can you conclude about its genotype and karyotype? Well, remember that if orange and black are carried on the X chromosomes, males should normally only inherit one. So they should either be orange or black. So you should not see normally a calico male. If you do see a calico male, that means they have the orange and black allele. They must have 2 X chromosomes and a Y chromosome. So they're aneuploid. They have 3 sex chromosomes. And that's the only way you can have a male calico cat. So again, X inactivation is epigenetic. We still have the extra X chromosome there, but it's been tightened into a bar body so that it's not expressed. When we go to make eggs and sperm, that bar body will get, or rather eggs, because the bar bodies are formed in females normally, that X chromosome will unravel, and either X chromosome can then pass to the offspring. So again, it's affecting my phenotype, but my offspring can inherit either one of my X's, regardless of inactivation. Genomic imprinting is another example of epigenetic inheritance. Rather than affecting a whole chromosome, it's usually a gene or a section of the chromosome. So a piece of the gene, or sorry, a segment of DNA, again, or a gene, is imprinted or marked in a way that affects gene expression. So imagine this is my whole chromosome. There's a section of it that's going to be chemically modified. So it's called methylation. And the methylation will make this part of the DNA become very tight. And again, when we make DNA be tight, it can't be used. We know now that imprinting occurs in many, many different organisms, including ourselves, and that it can affect a single gene, part of a chromosome, or even an entire chromosome. In many cases, it's affecting the gene of one parent. So this is a way that offspring are going to distinguish between maternally and paternally inherited genes. So instead of expressing both the mother and the father's copy like we normally do, they'll only express one. So one will get shut off by this methylation, and the other allele will be expressed, so that you only see either paternal or maternal inheritance. In the example we're going to look at, we see paternal inheritance. The mother's gene is imprinted. And again, remember, the imprinted gene is made tight and can't be read. So this is well studied in a gene called IGF-2. This stands for insulin-like growth factor 2, and it affects growth of the mice. And so the phenotype is size. And so if you look at this picture, you have a normal size mice and this cute little dwarf-sized mouse. And what they found is that normal and dwarf offspring can have the same genotype but different phenotypes. And this is because this is going to follow paternal inheritance. So if I look at the cross, I take a mother who's homozygous dwarf and a father who's homozygous normal, and I'm going to get a heterozygote. But the normal copy here, in this case, came from the father. So the normal copy came from the father. And if the normal copy comes from the father, then the offspring is normal. If I do the alternative cross, where the mother is normal and the father is dwarf, you still get a heterozygous offspring. So same genotype, but the normal copy is from the mother. And even though there's a normal copy from the mother, you will get dwarf offspring. This is because the father provided the dwarf copy. And because this is paternal inheritance, only the father's copy gets used. So what happens is in the egg, the IGF-2 gene is imprinted. In the sperm, it's not imprinted. So now when the sperm and the egg get together, the mother's copy is marked up and can't be read, but the father's copy is not marked up and can be read. And so the offspring can only use the paternal copy. So if the father gave them a dwarf gene, they'll be dwarf. If the father gave them a normal gene, they'll be normal. So this is one of the best examples of imprinting in mammals that we have. Imprinting is really, really fascinating to me. I call it the Wild West of biology, and I think this is an area where we're going to see a lot of research in the future because we're finding out that there's a huge impact of nutrition and environment in imprinting that seem to mark the DNA in ways that affect our risk for heart disease, diabetes, and cancer later in life. There's also, and I'm going to tell you an interesting example of both of these in a second, but there's also examples that we've now seen where the imprinting doesn't just last the lifetime of an individual, it lasts more than one generation. For example, nutrition in a grandparent might mark the DNA, and that marking might last into the grandchildren. So this is transgenerational. So one of the really interesting studies they've done on this is looking at the effects of famine. And they did this using, in countries where there was really good records. This was in Sweden, I believe. I think it was Sweden. And Sweden had really good harvest records. So they could predict famine based on harvest records. And this was going back to the 1800s that they could do this. And then they went into future generations, into modern times, looking at grandchildren, great-grandchildren. So notice I'm referencing multi-generations and harvest records, which is an environmental thing. And what they found was that if a grandparent experienced famine, then grandchildren and great-grandchildren were less likely to be obese and have diabetes. So in other words, if they had years of plenty, then grandchildren and great-grandchildren were more likely to be obese and have diabetes. So grandparents' food status, and particularly at a critical period in male development. So the famine or plenty is around 11 years old. So if the male around 11 years old experienced famine, then his grandchildren will be less likely to be obese. If around 11 years old he experienced plenty, then his children were more likely to be obese and have diabetes. And so the hypothesis is that famine and plenty mark important genes. They imprint important genes that control obesity and diabetes risk, or that affect obesity and diabetes risk, and that these markings persist at least for 2 or 3 generations. And this is just the beginning of some fascinating stuff they're doing in these areas. There's been other studies too. And I think it's really significant because it tells us that the things that we get exposed to can have huge impacts on not just our own health, but the health of our offspring and our offspring's offspring. And so that kind of changes our whole understanding of genetics, because it's not just mutations. It's these subtle changes in the DNA that can also affect health, our health and the health of our offspring. So I think that's an area to watch. I think we'll see lots of discoveries in it in the next 50 years.